Okay, so now you see my screen well?
Yeah, I do.
Only the presentation?
Yes.
Okay, so hi everyone. I'm Miri Neeman. I'm a doctor, physician, hematologist working at Tel Aviv Medical Center. And now I'm at a PhD at Yuval's lab here in Tel Aviv University. And I will tell you about our microarray technology that we use in the Sanguine project and show you a little bit of results. So just to a few minutes about epigenetics before we start. So even though, as we all know, we have exactly the same DNA in each one of our cells, we have different cells that exhibit very different functions. And this variation is controlled by epigenetics. This is chemical and physical modifications that do not alter the sequence of the DNA, but they control which genes will be expressed. And this can be regarded as the traffic light that places our genetic code and tells us which genes should be on or off and active or not. And although we have only one DNA sequence, in each of our cells, we have multiple epigenomes that correspond to different cell types. Here, just to show that there are three main epigenetic mechanisms. One of them is histone modification, the other is DNA methylation, and the third one is microRNA regulation. And I will talk briefly about the DNA methylation process because this is the epigenetic mechanism that the Sanguine project is based on. So if the genetic code has all the information our cells need for protein making, the epigenetic modifications add another layer of information. They regulate the exact and the unique combination of regions that turned on or off. This pattern changes between different tissues, different states of the cell, and they influenced by the environment and even with age. This mechanism enables fast adaptation to the environment. It reminds the evolutionary process, only it's really fast and doesn't need to take millions of years. Within the DNA, there are regions that are rich in cytosine base and then followed by a guanine base. And they called CpG islands. Most of the CpG islands are found in gene promoters. And when cytosine is followed by guanine, DNA methyltransferase enzymes can methylate the cytosine. They add a methyl group, and this change is preventing the translation of the methylated region. So the gene is turned off. And we have another group of enzymes, TET enzymes, that they convert the methylated cytosine back to the demethylated state. And then we see they put a hydroxy group on this cytosine, and this is one step in the demethylation process. So within the Sanguine project, we characterize the epigenetic patterns by fluorescently labeling those two modifications. And it is already known that every cell type has a specific methylation profile. The sperm and egg carry their methylation profile, and after fertilization, they shed the epigenetic information, and during differentiation and embryogenesis, they gain in again all the epigenetic program. And the heat map you see on the bottom is an important study published in 2009 by an Israeli group, actually. They managed to characterize different tissues by their methylation profile. This CpG modification can be chemoenzymatically labeled to measure the distribution of 5-methylcytosine, which is the methylated state, and 5-hydroxymethylcytosine. We use a chemoenzymatic labeling enzym that was developed in our lab. and it enables specific labeling for both modifications. This is an experiment that demonstrates stretched DNA fragment before and after 5-HMC. This is the hydroxy methylation before the labeling. Here you can see actually the 5-HMC modifications on the DNA molecule in red. And now I will briefly just explain how the labeling works. So first step is labeling the hydroxymethylated bases, the 5-HMC. And for this, we add an enzyme and a cofactor to the DNA. The enzyme binds the cofactor specifically to hydroxymethylated CpGs. And on the next step, a fluorophore is added and clicks to the cofactor. And now we have DNA with directly fluorescently labeled 5-HMC, like you see in red. And then the second stage is a different enzyme and different cofactor added to the DNA, to the labeled red DNA. And this one is the enzyme binds the cofactor to the unmodified CpG sites, meaning without hydroxy and without methyl group. And the fluorescent is added and clicked to the cofactor, which you see in green. So now we have genomic DNA with dual fluorescent labeling, red for hydroxy, 5-HMC, and green for unmodified CpGs. And after the hydroxy is taken by the first reaction and the unmodified CpG is taken by the second reaction, we can tell that unlabeled regions are the methylated ones. So 5-HMC is labeled directly, and 5-MC, the methylation, is inversely correlated to the labeled unmodified CpGs. Okay, so just to explain the DNA microarray technology, every spot on the microarray contains multiple strands of DNA, and these are called probes. The DNA sequence on each probe is unique. Thousands of probes are arrayed on a solid surface, usually it's glass, and the precise location and sequence of each probe is recorded in a computed database. The sequences of the probes are selected according to the purpose of the array. They can be regions of known genetic mutations to perform genetic screening tests, for example, or they can be sites that are prone to epigenetic modification and more. Microarrays can be large-scale discovery array with almost a million different probes. Usually, it's used to discover a wide range of data, or it can be a custom array with a limited number of probes that are selected specifically for genomic location that are relevant to a group of clinical entities that we're interested in. Next step is to hybridize the labeled DNA to the microarray. This slide here illustrates how labeled DNA strands of DNA can bind to the probes on the array. Very specific hybridization conditions are needed to make sure that the DNA that binds to the array has the maximal match to the sequence of the probe. It means that the hybridization is specific. This way we can be certain regarding the sequences that we interpret as methylated or unmethylated. It's mainly important if we want to learn about biological and molecular pathways using the discovered data. So here I will show our workflow in general. So we take peripheral blood drawn to the streak tube like you do in your site. The streak tubes here in the lab or in JAX Bio are centrifugated to separate the whole blood into the components of plasma and buffy coat and the red blood cells. And then genomic DNA is extracted from the buffy coat, the white blood cells layer, and the cell-free DNA is extracted from the plasma. This is in case of our sanguine project. And then DNA, like I talked earlier, is fluorescently labeled for the epigenetic modification that we're interested in, and then loaded and hybridized to the microarray. And after several hours, the microarray is washed and scanned. Okay, sorry. So here you can see real scans from first microarray experiments we did here on a commercial microarray with 60,000 probes. And each slide can contain 8 samples if there are 60,000 probes. Here we labeled newly diagnosed AML patients and DNA and healthy control that we collected from Tel Aviv Medical Center. So the 5-HMC, the hydroxymethylation, is in red, and the unmodified CpG is shown in green. On the bottom, on the bottom you see the same scan, only in zoom. And after hybridization of the sample DNA to the array, strands of DNA that has a fluorescent label on them will be visible after scanning. And the intensity of the spots represent how many labels are on the specific DNA strand. The epigenetic pattern between individuals and between different clinical states is very, very similar, approximately 99%. And we are looking for only the differences, only the probes that has significantly different intensity levels between 2 chosen cohorts. Like here, it's AML and control. This idea of finding differential probes allows us to design microarrays containing only small amount of probes, but only the meaningful ones. We don't need all the 100,000 probes that are similar between the cohorts. And this way, we lower the cost. We are allowing more samples to be loaded on a slide and make the microarray making process easier. And you can see here that in zoom out, the patterns look similar, but after zooming in, we can find some differences in the yellow arrows that you see, the differences in the intensities. And this heat map was created after analyzing few samples from the first experiments. And we see here we limited the results to find only 200 most differentially methylated regions. Each row represents a genomic locus, and the columns represent the samples, AML and healthy. And specifically here, we received 100% specificity and sensitivity, meaning that the differential probes we found were able to classify correctly all AML and all control cases. Of course, this is a very small cohort, just to learn the concept and not for biological conclusions. So, of course, the larger the cohorts will be, we will have stronger and more significant results. So now I'll talk a few minutes about the Sanguine project. It's about discovering a unique epigenetic pattern for several hematological malignancies. Our cohorts are plasma cell disorders, MGUS, smoldering myeloma, and active myeloma. Lymphomas, we have follicular lymphoma, marginal zone lymphoma, diffuse large B-cell lymphoma, and Hodgkin's lymphoma. And myeloid malignancies, we have myelodysplastic syndrome and acute myeloid leukemia. So the aims of the project are to discover unique epigenetic patterns and to design a hemachip using the discovered data that will successfully recognize all entities from peripheral blood samples. Okay, so I said that we are combining data from cell-free and genomic DNA. So just to explain, what is cell-free DNA? Dying cells release to the bloodstream fragments of DNA from their nucleus. So tissues with high turnover will have more representation, like cancerous tissues. In case of cancer, solid or hematologic, there are fragments of DNA originated in the cancer cells in our bloodstream, and this is far away from the tumor location. cell-free DNA also used to test fetal, for example, chromosomal abnormalities. It's taken from mother's peripheral blood. It's called the NIP test. And on the other hand, genomic DNA that extracted from blood or tissue will reveal data of the sampled tissue, blood cells, lymph nodes, solid tumors. In case of hematological malignancies, this is a special situation that many times we have the cancer cells in the bloodstream, which allows us an access to the genomic and cell-free DNA of the cancer cells. And our technology integrates epigenetic data from cell-free and from genomic DNA. So now I will show some results, preliminary results. It actually works, our technique. Part of the analysis, we combined a few cohorts, you will soon see, just to increase the size. So here we see results from genomic DNA of 26 AML and 26 control samples. The differentially methylated regions between the cohorts were identified, and these preliminary results, we see that a heat map with a hierarchical clustering, and each row represents a differentially methylated region. And you see the colors vary very much. And the analysis includes testing how each sample classifies when using the differential regions that we found. So 25 out of 26 AML samples were classified correctly, and all controls, 26 controls, also were classified correctly. Also, the heat map demonstrates clear distinction between the AML and control, as you can see. Second analysis we made here is results of genomic DNA of 18 diffuse DLBCL patients and 18 controls. Here we see that the analysis identified differentially methylated regions, and as we see on the heat map, they're very well distinct from another. And when we look at the classification, we see that 17 out of the 18 DLBCL was correctly classified, and all the controls were also correctly classified. Here are preliminary results for active multiple myeloma. We have here 14 myeloma and 14 controls. We see here the differentially methylated regions again, and we see a clear distinction in the heat map. All multiple myeloma cases were classified correctly with the differential probes, and also all the controls were classified correctly. Here we see the smoldering myeloma versus the control samples. It's 20 smoldering myeloma and 20 controls. And we see that all the smoldering myeloma were classified correctly, and 16 out of 20 controls also were classified correctly. And lastly, with plasma cells, this is for MGUS cohort versus controls. So in the MGUS case, we have 28 out of 33 MGUS classified correctly, and 32 out of 33 controls classified correctly. But just to remind that now we already have larger cohorts. This was made like a few weeks ago. And this is data from genomic DNA. It is not yet integrated with the cell-free data. Just to say that these classification results reflect the clinical and the biological nature of the three cohorts. It's interesting to see that we could accurately classify the entities active myeloma was best classified, and then smoldering, and lastly MGUS, just because it's less active states. Here we also made an analysis of finding differential probes within the different plasma cell pathologies itself. So myeloma is the most active disease, and smoldering myeloma and MGUS are less active states. So we see then we see here that when we test the samples classification according to the differentially methylated regions, on the left we see the classification based on myeloma and smoldering myeloma. 13 out of 14 multiple myeloma cases were correctly classified, and 18 out of 20 smoldering were correctly classified. And in the middle, we see the differential regions of multiple myeloma and MGUS. And here, all of them were classified correctly. And on the left, we see the distinction between smoldering and MGUS. And here we see the least accurate results out of all the three examples. So next, here are more results of differentially methylated regions between different diseases. You see here DLBCL versus AML. We took 18 DLBCL and 26 AML, and all DLBCL were classified correctly, and 25 out of 26 AML were also classified correctly. So we see that we not only can distinct the disease from healthy control, but also between the different malignancies. And last one also, like the previous slide, these are differentially methylated regions between DLBCL and between multiple myeloma samples. Here we see 18 DLBCL versus 14 myeloma, and 16 out of the 18 DLBCL were classified correctly, and all 14 multiple myeloma were also classified correctly. So actually, it's a bit shorter than I thought, but it's okay because we lost 5 first minutes. So I'm done, and I can answer any questions if you have.